Hello, chemistry students. This is Professor Sean McMahon, and this is Chapter 9, our 4th lecture. We've finished covering our topics with the different types of solutions and why solution chemistry is so important when studying chemistry. We talked about the different types of calculations with percent concentrations, mass to mass, mass to volume, volume to volume, and what's really important is molarity. The reason why molarity was so important is we could tie that into our solution chemistry or our solution stoichiometry, because so many reactions take place under aqueous conditions, essentially being dissolved in water. That goes with our double displacement and single displacement. So for this last portion of the lecture, we actually continue talking about solutions, but we kind of expand our discussion and talking about different types of homogeneous mixtures and heterogeneous mixtures, because after all, a solution is only one type of homogeneous mixture. So that's what the remainder of this chapter is. There aren't any necessarily activities. This is more of conceptual understanding. This is really geared more towards allied health students than general chemistry students, or your classical science students who take chemistry like biology or physics students. This is geared more towards specifically allied health. So let's go on and take a look. So just as a reminder, some properties of solutions we have to take into account is the particle size, the distribution, the polarity, because that's going to help us determine whether or not it dissolves in water. Remember, like dissolves like. Water is polar. So if you're a polar covalent compound, or extreme polarity would result in ionic, because there wouldn't be uneven sharing of electrons. It would basically be a complete electron transfer. So ions and polar compounds dissolve in water very well. So let's further classify some things. So in a solution, it's a little bit different than we'll see colloids or colloidal dispersion. So you may have heard this. I'll get into the different types. And then there are also what are referred to as suspensions. We've actually dealt with suspensions as well, but I'll go in greater detail. So first, in a solution, the solute dissolved is typically extremely small, and it evenly distributes. That's part of a homogeneous mixture. It's an even distribution throughout. So when you think of the particles, you can think of tiny things dissolved in water, like ions, right? So your ions are small. The solute and solvent, you can't visually see them. And when you look through the solution, it is uniform throughout, okay? So you can't really see the solute particles or the solvent particles, and they're typically transparent, okay? The solute particles are so small that they actually can go through a filter or a semi-permeable membrane. So what do I mean by semi-permeable membrane? It's a membrane where there's such small pores, only very tiny particles can fit through. When you think of filter paper, that's a macro thing. So the pores are actually really large compared to a semi-permeable membrane. An example of a semi-permeable membrane is a cell membrane. Okay, so with solute particles, they're so small they can go through cell membranes. So you can think of like sodium and potassium. They can easily flow through cell membranes. So semi-permeable membranes allow solvent and these small solute particles to pass through. They pass through filters because those gaps in the filter are so large, they can easily fit through that. So the way you would separate the solute from the solvent in a solution, which is a homogeneous mixture, is you would have to use evaporation or distillation. You would have to boil the water off. That is the way you could separate them. Now, colloids are a little bit different. Those solute particles, they're bigger than small molecules like glucose. Glucose is only a 6-carbon structure, or sodium chloride ions. Colloids, those particles tend to be, for example, a vitamin or a protein. They're much larger particles. You can think of them as macromolecules rather than just small little molecules. So like your lipids, your DNA, those things, they're a little bit larger. Now, it's still a homogeneous mixture. There's no settling. Typically colloids, because they're homogeneous, they're uniform throughout. It's just that the particles they're bigger. They're still pretty much naked to the human eye, but they're larger particles. They can pass through a filter, but they cannot fit through a semi-permeable membrane. So you would need, for example, some type of protein pump or channel. So an integral membrane protein would allow these larger particles to come out. So what are some examples? Well, there's several different examples of colloids. I always like to kind of focus for students to help them see. Again, you could have, it is a homogeneous mixture, just like when we talked about solutions. You can have examples of, liquid and gas, gas to liquid, gas to solid. But I'm going to do one that I think is fairly easy for people to understand, is homogenized milk. So when you think of milk, if you look at it, it's uniform throughout, right? For the most part, it's one color. You can't see the actual particles of solute or solvent, but we know that there's fats that are dissolved in there. There's amino acids, there's sugars. So they have large particles. Now, eventually, if the colloid milk spoils and it begins to settle out, at that point, it is no longer homogeneous, it becomes heterogeneous. And we'll be getting to that next, because that's essentially what your suspensions are. Another example of a colloid is blood plasma, right? We have, in blood plasma, you have things dissolved that are rather large. You have red blood cells. You have, again, proteins, lipids. So this is considered a colloid. It's uniform throughout. With suspensions, these are heterogeneous. They're non-uniform. That's typically when you have really large particles and you can actually see them with the naked eye. So these not only get trapped in a semi-permeable membrane, like the cell membrane, but they would even get trapped with filter paper. So in prior discussions, when we were talking about double displacement precipitation reactions, we started with solutions, but when we combine them, we produce a precipitate, which becomes basically a heterogeneous mixture. We have this suspension, the particles are suspended, and then we could easily, through centrifugation, separate them by decanting the liquid above the precipitate, or we can use filter and use the filtration techniques that we've talked about in prior lectures. If you just had a suspension and you could see the particles, eventually it would settle down and precipitate to the very bottom of the container, okay? With suspensions, you have to kind of stir it to keep the particles suspended, otherwise they will settle out. So you can think of, like, salad dressing, for example. So examples of suspensions: kaopectate, calamine lotion, antacids, liquid penicillin. Again, a lot of medications are suspensions. They have large particles. They're suspended in solution. You have to shake them before you ingest them to try to even them out because they're non-uniform. You let it sit, it will precipitate out to the bottom. So that's why a lot of times with suspension medication, you're asked to mix it to try to spread it out a little more evenly. So the big takeaway when we're comparing solutions and colloids and suspensions, just remember solutions and colloids are homogeneous. The suspensions are hetero. Okay, so your suspensions are heterogeneous. These are easy to separate. We can just filter. Okay. Now, filter paper pores are too big. So with the homogeneous mixtures of both colloids and suspensions, right, these are homogeneous, you can't separate the particles through filter paper. With colloids, because the particles are big, they're macromolecules, I can use a semi-permeable membrane like the cell membrane, and we can talk about the concept of dialysis in a few moments. We'll be talking about how dialysis is an example. Cellophane membranes or semi-permeable membranes allow us to separate proteins from in colloids. Now with true solutions, because the particles are so small, this is where you're going to actually have to use distillation to separate them. Okay, so those are the ways. Another way you can think of it is solution as the smallest particles. Ions are small molecules like glucose. Colloids, they're still small. We can't visibly see them, right? They're to the naked eye, you can't see them, but they're larger molecules. So I like to tell people, think of vitamins, proteins, but both of these do not settle because they are homogeneous. Okay, so we already talked about this. They are evenly distributed. Now with suspensions, since the particles are so large, you can actually see them, and these rapidly settle, okay? So this is an example of your heterogeneous, and this is why we can use filter paper, okay? So earlier, when we were talking about solution chemistry, we brought up electrolytes and non-electrolytes. The reason why electrolytes are considered solutions is because electrolytes just basically means I have ions dissolved. If an ion is dissolved in solution, or if an ion is dissolved in a solvent, it's going to make a solution, which is a homogeneous mixture. Those are the smallest particles, okay? Non-electrolytes, right? They can also dissolve as molecules as long as they're polar, okay? So we've talked about that. So let's kind of look at it in terms of, will this, as a review, dissociate, or, you know, what type of electrolyte will this solution be? So here's an example of ethylene glycol. It's polar, so it can dissolve in water, but even though this says aqueous, because this is a covalent molecule, it will not break apart into ions. And ethylene glycol is basically, you can think of it as this structure. There's 2 carbons, and then you have 2 alcohol groups, your OH groups, and then the remaining hydrogens. Okay, so this OH group makes this compound polar, and it allows it to dissolve in water. That's why we have aqueous. But because it's a covalent compound, it does not break apart into ions. So 1 mole of this molecule gives me 1 mole of dissolved solute, okay? And this is a very small molecule compared to macromolecules. Now, if I throw calcium chloride in water, well, this is ionic, I know, because I have a metal and a non-metal. So what is it going to do? If I throw it in water and it's soluble, solubility rules, we know it's going to dissociate to its respective ions. So now I have 1 mole of calcium ions, but I have 2 moles of chloride ions. So 1 mole of the calcium chloride electrolyte essentially gives me 3 moles of ions. The only reason why I'm bringing this up is to help us kind of reconfirm what we learned earlier with strong electrolytes, weak electrolytes, non-electrolytes, and just to understand the dissociation of these ionic compounds in solution. It also shows the concentration of ions. So here's another example where they're trying to show the effects of the concentration, where, ethylene glycol, because it's a non-electrolyte and it is basically a molecule, I only get in 1 mole of ethylene glycol dissolved in water, 1 mole of solute. If I have 1 mole of sodium chloride, it can split apart to 1 sodium ion and 1 chloride ion. So I get 2 moles of solute particles per 1 mole of sodium chloride. And I get in 1 mole of calcium chloride, 3 moles, right? Because there's 1 calcium ion and 2 chloride. I get 3 moles of solute particles there. Both of these are considered strong electrolytes because there's 100% dissociation, and these are soluble salts. This is considered a non-electrolyte because the ethylene glycol, if you remember, it was C2H6O2, is a covalent compound, and it doesn't dissociate. We see the effects because as there are more solute particles dissolved in water, the boiling point rises because it's harder to boil it, okay? So the other topic that we want to talk about, since we brought up solutions, electrolytes, is osmosis. Because again, osmosis is essentially the flow of solvent. Now, how does it flow? It goes from a less concentrated solution to a higher concentrated solution. Now, people get confused because they think of diffusion as high concentration to low, and that's true with particles. But with water, it's kind of the opposite. Water wants to dissolve solute. So if I have a container and there's, let's say, a semi-permeable membrane, and there's more particles on one side compared to the other, the water is going to want to flow to the side that's more concentrated to help dissolve and surround those solute particles. Remember the solvation that we had talked about earlier. So that's what osmosis is. It's the flow of water to where there's fewer solute to where there's greater solute. And the reason why they want to go where there's a greater concentration is they want to help dissolve it. But you need a semi-permeable membrane. Okay, so semi-permeable membrane has very small pores, and that's going to allow the water to flow. Okay. The level of the solution with the higher solute concentration as a result rises because as the water flows to help dissolve it, the solution itself is going to rise. The concentration of the two solutions on both sides will end up becoming equal over time if there's enough room for the water to rise. So here's a better example of the drawing than what I did. Is essentially on this side, we just have the solvent water. On this side, we have sucrose, which is fairly small. It's a disaccharide. It's C12H22O11. And because it's completely polar, it dissolves. It's also a covalent compound, so it doesn't dissociate into ions, but we could dissolve sucrose in water. Now, per 1 mole of this, 1 mole of it will dissolve. Now that I have the solute sucrose in here, it needs to be surrounded. That's part of what dissolving is, by the solvent. So essentially what's happening is the water is going to flow to help surround solvation, those solute particles. As the water flows in the direction of the more concentrated solution, what will happen is this water level rises, and you can see that here. Okay, so there's water going towards where there's more solute, more concentration, and as a result, the water level or the solution height rises. Okay, now what does that do? That creates greater osmotic pressure. Now, this is confusing for people, and I get it, but essentially, if you can think of as pressure as force per unit area, kind of pushing down, you can think of it as you jumping on a scale, and there's, force per unit area of that scale. As the water flows to where there's more solute, right? So if we have a semi-permeable membrane and I have solute dissolved on one side, the water is going to flow towards the side that has more solute. Eventually, this water level or solution level is going to rise. And what will happen is there'll be a difference in the heights. Now, that difference is what your osmotic pressure is. When there is a greater height, there's more pressure due to gravity pulling down on that difference in height. That's essentially what osmotic pressure. I used to try to tell students, you can think of osmotic pressure as, the water is being pulled towards where there's more solute particles. And as a result, because the level rises, there's a greater osmotic pressure. So increases as the number of dissolved particles in the solution increases. So more solute, greater osmotic pressure, because you have to have a greater rise. So how is this relevant to us and how does it apply? Well, osmotic pressure can be seen with red blood cells. Okay, so typically your red blood cells, like other cells, have semi-permeable membranes. That's essentially what your lipid bilayer membrane is. It's semi-permeable. So to maintain an osmotic pressure that cannot change without damage occurring, you got to make sure that whatever's going in your body is at the right concentration. So you got to maintain an equal flow of water between the red blood cells and the blood plasma, right? So the red blood cells are dissolved in your plasma, which also has white blood cells, proteins, right? And several other things as well. Fibrinogen for scarring and things like that. So now, if I have a solution that has the same concentration as a red blood cell inside of its red blood cell, then it's considered to be isotonic. Iso means the same, right? So like if I have an isosceles triangle, I have two congruent sides. The sides are the same. So an isotonic solution exerts the same osmotic pressure as red blood cells do, okay? So we have to make sure, if we're in the allied healthcare, that any IV that goes into the bloodstream intravenously is at the same osmotic pressure as a red blood cell. It has to be an isotonic solution. And to have that, and this is why we reviewed percent concentrations, I need a 5% mass to volume glucose solution. What does that mean? I have 5 grams of glucose per 100 mils of solution, okay, because it's mass to volume. I also have to have a 0.9% mass to volume sodium chloride, because again, your cells, right, they're electrolytic. There's ions dissolved, right? We need ions. So you would have 0.9 grams of sodium chloride per 100 mils of solution. So that's what your IV bags are essentially, right? They have to have these concentrations. So a red blood cell placed in this solution keeps its volume. Why? Water doesn't flow in or out in a greater extent. Now, because it has a semi-permeable membrane, water can flow in and out, but they're going to go in equal movement. There's not going to be a pull in either direction. So it basically maintains its size. Hypotonic is where you have a less concentrated solution. So it has a lower osmotic blood, pressure than a red blood cell. So you can think of it as, if I have a solution that's just mostly water and I put a red blood cell that has, particles dissolved, what'll happen is the water will rush in to try to, dissolve that solute. So as water's rushing in, the water level rises, the cell's going to expand. And unfortunately, when water flows into a cell, hemolysis, lytic, it's going to burst and you're going to kill the cell. So that's not a good thing. We don't want that to happen. With the opposite situation, you could have what's called a hypertonic solution. Just think the term hyper means more, like that kid's really hyperactive. It means they have a lot of energy. Well, a hypertonic solution means I have more solute dissolved in that solution than the red blood cell. So if I have a red blood cell and it has a certain concentration, and then I put it in a solution that's far more concentrated, now what's going to happen through the semi-permeable membrane of the red blood cell, the water is going to want to rush out to dissolve that really concentrated solution. So as the water flows out, now you're going to cause a shrinking in size, which is referred to as crenation, and that's not good either. An example of this is brining, right? If you take a cucumber, which has semi-permeable membranes because, it has cell membranes, when I put that in a salty solution, even though the salty solution goes into the cucumber, a lot of the water goes out, so it kind of shrinks, right? So we have the solute going into the cucumber because it's semi-permeable and salt is small enough to go through the semi-permeable membrane, but the water's rushing out of the cucumber, and that's how you get a pickle. So here's an example of, the red blood cells in your isotonic, hypertonic, and hypotonic. Hypertonic is more concentrated than red blood cell. Hypo is less, okay? So in an isotonic solution, the water goes back and forth. The cell, because it's the same concentration, keeps its size. In a hypotonic solution, the red blood cells actually are more concentrated than the surrounding solution. So now water rushes in, and this is where it can swell up and even burst, hemolysis. In the hypertonic solution, there's a greater concentration outside of the red blood cell, and we see that the arrow is bigger. So the water is rushing out and it begins to shrivel, crenation, and that can cause death as well. So that's why it's very critical if you're in the allied health care field and you ever provide any type of medication or IV intravenously, you have to make sure that it is an isotonic solution. Now, we've brought up the concept of semi-permeable membranes with red blood cells and just cells in general. So we also talked about when we have homogeneous mixtures, solutions, for example, the salt can cross not just the filter paper, but also the semi-permeable membrane. With colloids, and you have really large particles, they can't pass through a semi-permeable membrane. Okay, they can pass through filter, but not a semi-permeable membrane. So in dialysis, when we deal with kidneys and a nephron, right? The nephron, basically, you have the glomerulus, which filters the blood. And what's happening is, because this nephron has a cell membrane that has a semi-permeable membrane with small pores, you see that the solvent and the small solute particles can pass through the cell membrane. Now, I'm putting artificial membrane because I'm going to relate this to dialysis patients who don't have functioning kidneys and why they need to get dialysis treatment, okay? Large particles, though, like proteins and things like that, red blood cells, aren't going to go through into the nephron. And that's good because eventually you see with the nephron, the urine, it collects and then it goes into the ureter. And we don't want to excrete large particles like red blood cells or proteins, right? Okay. So waste particles are essentially what we want to pass through the semi-permeable membrane. So waste particles like urea, which is just an amide. It's like the simplest amide possible. It's basically this. So that's a relatively small particle. It's polar, it dissolves. But you want to remove this from the blood, right? Because it's a waste product. It's a way for us to get rid of the amines in blood from deamination with protein breakdown. And large amounts of amine aren't good because amines are weak bases. So we need to get rid of any excess. So we do this by filtering our blood and using our nephron, okay? So if you have a kidney that has failed, or we need to basically perform what's referred to as hemodialysis. We need to do the job of what the kidney would do. So how would we do that? Well, if the kidney fails, you have to go and you have to have your blood filtered. So you're hooked up to a hemodialysis machine, and it passes through, and you see that the blood goes through basically a coiled semi-permeable membrane structure, and it's placed in the coiled filter is placed in a very low hypotonic solution where it's referred to as the dialysate. And what'll happen is now your waste products can go through the semi-permeable membrane and go be essentially pulled out because it can pass through. So as a result, you get rid of the urea and other waste products as it goes through the system, and then it returns through a return phase to the patient, and now it's clear of the urea. And you can see that it actually has these coils. It goes through this. I don't know if anybody ever donates blood. It's kind of similar to if you ever do what's called a double red blood cell donation. That's typically what I do, where there is a in phase where the blood goes in, they kind of centrifuge it, and then there's a return phase where it comes back. It's actually really interesting because it comes back kind of cold. It's not body temperature, and you can actually feel that. So it's interesting. This is a very long process. With a double red blood cell donation, it's usually about 20 minutes. This is typically a little bit longer than that. But anyways, hopefully this helps you understand why we study chemistry, because we can apply it to biology, the medical field, and so many other areas. I hope this helps you understand that solutions are only one type of homogeneous mixture. We saw earlier that colloids, like blood, are an example of another homogeneous mixture with small particles that we can't visibly see, but through semi-permeable membranes, we can actually separate components of it, the solute particles. And then we also talked about suspensions as well. So hope you appreciate the video, and thanks for tuning in, and I'll see you next time.